Here, we are going to discuss the simple topic from the chapter of human reproduction, that is infertility. Infertility, this condition can be defined the inability to produce children in spite of unprotected sexual cohabitation. In the present world, there are many technologies available to avoid the problem of this infertility that will be termed by the word assisted reproductive technologies. Under this, right now we are going to discuss a simple technique that is called IVF. It will be expanded in vitro fertilization. Generally, we know the fertilization in human body occurred inside the body. Meantime, in case of IVF, it occurs outside the body. So, the particular couple, men, female as well as male, their gametes will be collected and fused in the in vitro condition. Maybe that fusion can be happen in a test tube. So even the IVF can be termed by the other word test tube program, test tube baby too. So once the fusion occurs, it will give a zygote. Meantime, this IVF successful rate is just 20% only. The zygote which is formed, then it will be allowed for the further cell division. Due to that, they will be form a embryo also. So the cell division will be developed up to the stage of 8 blastomere stage. Once the 8th blastomere stage is reached, then those collection will be taken in a injection and then it will be allowed inside the body. So by following this IVF, the technique what I said just now, it is called embryo transfer. Particularly if you want to term in a embryo transfer, the stage of 8 blastomere cell transfer is called ZIFT, zygote intrafallopian transfer. Meantime, in case the 8 blastomere stage is overcrossed, then we need to call the term IUT, intrauterine transfer. Naturally, we know that the formation of zygote which occurred in a place of oviduct, that will be followed and finally it will be reached to the place of uterus. So zygote formation in oviduct, further the development will be occurred, finally it will be attached on the endometrium wall where the baby will be developed further. Alright, so that is what, once the stage is crossed, of course the final place must be uterus. That is what in IUT, embryo with more than 8 blastomere cell should be in a uterus. So this is called IUT. The next famous method is GIFT, gamete intrafallopian transfer. This method is generally used in females who cannot produce ova but can provide suitable environment for fertilization and further development of embryo in the oviducts. In such cases, ovum from the donor female is surgically removed and then is introduced into the fallopian tube of such females. Such women then accept sperms from her husband during copulation. This is called GIFT. Next one is ICSI, intracytoplasmic sperm injection. This is another specialized technique to form an embryo in the laboratory in which a sperm is directly injected into the ovum placed in a nutrient broth. The zygote or early embryo is later transferred by ZIFT in women. The last method is called AI, artificial insemination. In some books it may be given AIT2, artificial insemination technique. This technique is used in those females where the husband either is unable to inseminate the female or has very low sperm counts in the ejaculation. In such cases, the semen of the husband or donor male is first collected and then is artificially introduced either into the vagina or into the uterus of the female, right? So these are the major method which we need to discuss over here. Even I have given one more term, intrauterine insemination. The last term what I explained just now, exactly if you want to term the introduction of the particular semen in the uterus region, then we need to use this term IUI, intrauterine insemination.